factors involved and their functions. And remember, it occurs in 3 different stages.
Yeah, so we're fixed.
So,
Can you write it down?
huh? First of all, let me find out what you actually know about it, then I can tell you what you might need to know about it. So, we're looking at transcription from DNA to RNA. It happens in 3 different phases. What are the 3 different phases?
The initiation, elongation, and termination.
Okay, so you have initiation, then you have elongation and termination. Initiation starts with recognizing of the portion of DNA that's going to be transcribed. What recognizes that portion?
The RNA polymerase and signaling factor recognize specific promoters.
Promoters, yeah. And so you have a promoter. And then you have the genes that are going to be transcribed, right? And so you have this RNA polymerase, right? And RNA polymerase contains a sigma factor. So this is RNA polymerase. This is your sigma factor. And so this first step is going to be your initiation. And so your RNA polymerase with the sigma factor recognizes the promoter. What is the job of the promoter? Well, the job of the RNA polymerase to bind onto the promoter. But what is the job of the promoter? If I'm going to promote something, such as going to a
Recognize?
okay,
To recognize.
it's a recognition of what?
What, like which sequences?
Which direction? Okay, so this promoter could recognize, could have a preset direction for that gene that's going to be transcribed, or it could be the gene on this side, so it could be going upstream or downstream, is what we call it. So the RNA polymerase, it recognizes the promoter with the help of the sigma factor, so it knows to bind down to that promoter, and then it will start to move down this DNA strand here. So it will move in this direction. And so that's the initiation portion. What about the elongation? What's going on with elongation?
Is that where it reads the DNA?
So it's reading the DNA, and so what is it doing?
It reads it and then it adds the RNA nucleotides along with
Okay,
it.
so I have the RNA nucleotide. And they are going to be added onto here, so you're going to get this elongation of RNA. Okay?
So the RNA nucleotides read the.
No, the RNA nucleotides are going to be added to
So,
this.
but the RNA polymerase reads the
RNA polymerase reads the DNA and adds the appropriate RNA nucleotides to make the new RNA. So it's like building blocks. If you're putting pieces of your Legos together to make a long chain of them, have to do it here. Except that they're in a sequence that is complementary to what the message is there. So this is your elongation here. So it keeps reading it all the way till it gets to this terminal point, right? And so at termination, after it reads all the way through, elongates this new RNA molecule, it reaches this point here, and you have termination.
Which it just terminates the molecule.
Yes.
The RNA.
So the newly formed mRNA is now going to be used, right? But what we know about prokaryotes is as soon as it's being made, it's being used, right? As soon as it's being made, ribosomes are jumping on there and making the proteins. And we know that because this RNA is going to degrade after it's been used. So it's no longer going to be around, right? So once it reaches here, the RNA polymerase will break off of there and stop elongating, therefore stopping the whole transcription.
So the RNA polymerase reaches the terminal region and
Mhm.
terminates?
Yes,
Or
it terminates
just terminates?
transcription. It goes off and does something else, whatever it needs to do.
So can you read it out?
Huh?
So can you read it?
Write it up. No, we gotta get through this stuff.
It's okay, we have time.
No,
We don't have lab today.
Huh?
We don't have lab today, so you just keep going.
No,
all the
we
time.
cannot do that either.
You can.
No, some of them.
You're like, we don't have money. We're not you.
So if you would take your notes as I was speaking, then you would have it written out, right?
So now we're on seven.
RNA polymerase, sigma factor, recognizes the promoter. After recognition of the promoter, the RNA polymerase will read the genes that are on the DNA. As it reads the genes in the DNA, it will add the appropriate RNA nucleotides to the growing RNA molecule. That is the elongation phase of it. The RNA polymerase will continue reading until it reaches the terminal region. Once it reaches the terminal region, it will cease to perform transcription anymore and will move away, and therefore ending transcription. That's it.
I lost you, but okay. I lost you after recognition.
I thought I had you at initiation. That's a Jerry Maguire. That's a Jerry Maguire reference there. You ever seen Jerry Maguire? You guys gotta get out somehow. Okay, now we're going to talk about describing in detail the processing of a eukaryotic mRNA.
This is hard.
This is not hard, it's
Well,
just
it's hard to
different.
put it in writing.
So, you have your nucleus, your pores. This would be a 4, right? And so, since eukaryotic cells are more complex than your bacterial cells, you have to go through some processing when it comes to the mRNA, right? So, after you have this transcription that goes along, you have what's called a pre-RNA. Pre-RNA. And that pre-RNA is made up of what? Two things that it's made up of. stands for
Okay, is that
exon
the exon intron?
and introns, okay? Exons are what?
Useful,
These are useful.
Wait, what's it made up of again?
exon and intron.
And introns are what?
Junk.
Junk or non-coding. Okay, so we have to process this out. So what has to happen to this molecule for it to be processed? I'll draw it down here again.
Is that where it's, now is that read by the RNA polymerase?
Not yet.
Okay, what
Oh,
about
well,
the
it's already
splice
been
of-
read. The RNA polymerase, this has already happened.
Okay, now what about,
Okay.
what is the splice of
what?
video?
I'll talk about that. Okay, before this happens,
I noticed
you have 5' cap and a poly A tail. That has to be attached to it, right?
that.
The 5' cap and a poly A tail have to be attached. So now, what happens next? Now we have this,
Splicing gets rid of the junk? Something gets rid of the junk.
Spliceosome.
I know, that's splices.
That's kind of what it looks like in real life, but in the 3D image it's different. Spliceosome does what?
Gets rid of the junk.
Yes, it comes through here, reads through here, and gets rid of the introns, since they are non-coding.
Reads where?
It reads this and gets rid of,
Well, what is this called? Pre-RNA.
this is your pre-RNA. So, that's going to leave us with only exons. Exons are coding, introns are non-coding. Okay?
No, exon are helpful, intron is junk.
Okay.
Exon is helpful, right? Exon is
The
good.
exons are useful bits of information. The introns are non-coding bits of information.
I just put it.
So this processing has to occur. First you have your 5' cap that is placed on here, and you have your poly A tail that is also attached onto there.
We have to find what?
5'.
Now, calling like that first when you start out, is that like your DNA template?
No.
Or what do you call that?
This is RNA.
Okay, just your RNA.
All of this is RNA.
I'm trying to figure out how
I
to-
already did the transcription. It's already occurred. We're talking about a eukaryotic cell. This is prokaryotic because as it is coming out, it's being used. But in the eukaryotic cells, such as the cells that we contain, you have to have processing before it can come out and be used. So we have this pre-RNA that is made. In order for it to be processed, you have to have a cap put on the 5' end and a poly A tail put on the 3' end. RNA is also read 5' to 3' just like DNA. After the spliceosome has come through and got rid of the introns, you're left with only the useful coding DNA, which is the exons. Okay? The purpose of the 5' cap and the poly A tail is to keep them together so that when the introns are taken out, it stays together. Okay? So it doesn't just go all over the place. Now, I have a processed mRNA. It will then